It is my pleasure to introduce Meghan Duffy. She has some really cool research that is actually, we have a lot of interest in and we've dabbled in a little bit, looking at the evolution and ecology of host-parasite interactions using the Daphnia model system. She is now an associate professor at University of Michigan. Very prolific publication record and a steady stream of NSF grants, including the prestigious, not just career, but the presidential career. So the very top echelon of the career grants. So very excited to have her here today to talk to us about her research. And today, for this seminar, she's going to be talking about the value of basic scientific research and using her model system to approach this, something that I know a lot of us think a lot about. So, yeah, well, I'll let Meghan take it from here. Thank you.
All right. Thank you, and thanks to all of you for coming. I've really enjoyed my visit so far, and I'm looking forward to meeting more people tomorrow. Before I get started, I want to acknowledge the co-authors on the work that I'll be talking about today. So the bulk of this work was done by Crystal Sanchez, who's a grad student in my lab, and this, the work I'm presenting today was part of her master's thesis. She had a lot of help in the lab from Naomi Huntley, who is an undergrad who did a really great job in the lab. And one of the really nice things about working on the system we do, so Daphnia, is that it's generally pretty feasible to spin off projects that work really well for undergrads or to involve them in our work. So we have lots of undergrads in the lab, and we really like that. And then Crystal was co-advised by me and Mark Hunter for her master's thesis. And so the chemical ecology expertise definitely came from Mark instead of from me. Okay, and then I've started this year doing territorial acknowledgements, and someone asked me about this before, so I'll explain a little more. The general goal is to acknowledge that we were not the first people to be here, and to acknowledge who were the first people to be here. And so you can go, if you're curious about this, for places you're visiting. There's a website that is a very useful resource. And so here, there are the Shoshone-Bannock tribes. And their ancestral lands covered present-day Idaho, Oregon, Nevada, Utah, Wyoming, Montana, and into Canada. So really a very impressive range. And then the Eastern Shoshone tribe has lived in the Wind River mountain range for about 12,000 years. I mean, that number just really is kind of mind-blowing to me. And this picture shows the Lewis and Clark expedition with Sacagawea, who is an Eastern Shoshone woman who was only about 17 at the time that she led them through and carried an infant with her. So we think of them as these really tough explorers, and she was a 17-year-old carrying her infant. So. Okay, so we're going to start with a quiz. And so what percentage of the U.S. federal budget is spent on non-defense research and development? All right, so we'll do a show of hands. I won't make you close your eyes. So I practiced this with my lab. I made them close their eyes, and then they said the numbers. I was like, no, that's not how it works. So who thinks it's less than 2%? We have a well-informed room. Who thinks it's roughly 2 to 10%? Few optimists in the room. 10 to 25%? Anyone in a really optimistic world where it's over 25%? Yeah, that would be a nice world. So the most recent number I could find was from 2014, where it's 1.6% of the U.S. federal budget is spent on non-defense research and development. And one thing that I think is really interesting is there was a study done recently that found that, so people really, they like science. They realize science has made their lives better, but they think we spend a lot more money on science than we actually do. And once you note how little of the federal budget actually goes to science, then there's a notable increase in the support for increasing funding for science. So I think one of the take-homes from this is that we need to do a better job of pointing out how little money we currently give to non-defense research and development. Okay, and so to try to help make this point, I made some bad bar graphs here, but so the total height of this is the total U.S. federal budget, right? And here's our tiny little thing that you can barely see there. That's 1.6% of the budget for non-defense research and development. If if we take this 1.6% and blow it out to be its own whole bar, and then we look at the different agencies, so this includes money that goes to NIH, the USDA, NOAA, most of it goes to those other agencies, and only 9% of that 1.6% goes to the National Science Foundation. So this is a small fraction of a really small fraction of the total budget. And why I'm specifically highlighting the National Science Foundation budget is NSF is the agency that has the mandate to support basic scientific research. And to make sure we're all on the same page with that, basic research is research that has no immediate application. And so today I'm going to tell you about basic research that we've been doing in my lab that's led to some unexpected results and that might actually end up having direct applications. And the take-home message will be that a drug that might help us fight fungal infections in people might be produced by the phytoplankton that are living in the surface of this, right below the surface of this lake. And we never would have found that if we weren't doing our basic research. Okay, so to introduce you some more. So research in my lab focuses on Daphnia parasite interactions. Daphnia are tiny shrimp-like creatures that live in lakes. They're really cute. Most people don't know about them, but I think they're beautiful organisms, and they're really common we have many lakes in Michigan. It's really great to be an aquatic ecologist in Michigan. We've got tons of lakes around, and they're chock full of these Daphnia. And so some of you may have had experience, I mean, some of you have actually worked with Daphnia. So, but some of you who haven't may have had experience with them. They're fairly commonly used in labs where you add some sort of chemicals to water and then you look at their heart rate or something like that. So that's where some people have had exposure to them. But so we work on Daphnia in my lab because they're really important ecologically. So they're the main grazer on algae and other phytoplankton in our lakes. So they help keep our lakes from looking like pea soup. And then they're a really key link in these aquatic food webs because they're a key resource for fish. So young bluegill sunfish have their stomachs just full of these Daphnia. So they're really important in lake food webs, and that's part of why we work on them. But we also work on them because they've emerged as a model system for understanding the ecology and evolution of infectious diseases. And so we've used them to try to understand why disease outbreaks begin, why they end, why they're as severe as they are, a lot of different things. And so they have many advantages as a study system, including their small lake populations have billions of them, so we can sample them and not cause problems for the populations. You can work with them in the lab and in the field. And then one that sounds sort of silly but actually is important is that they're transparent. And so we can actually readily assess whether they're infected and what they're infected with. And that ends up being a really handy thing about the system. Okay, so one of the things that we've done some work on over the years is looking at how the diet, the foods that Daphnia feed on when they're exposed to parasites, how that alters the outcome of infection. And this is something we'd worked on a bit in the past, but that we started really working on when Crystal joined my lab a couple of years ago. So here I am with Crystal and another guy at the March for Science last year. And so this again was like Crystal's master's thesis work. And for this work, so most of us don't give a whole lot of thought to what organisms eat, even if we're ecologists. We tend not to think about this too much. And if we do, we tend to think about foods in terms of their nutritional value. So is it a nutritious food or not a nutritious food? But diets, yes, they have to satisfy the everyday metabolic requirements of organisms, but they also can serve other roles. And one of those other roles is that sometimes diets have medicinal properties, and I'll talk about this more in a little bit. But it's also important to keep in mind that many foods contain toxins. And the old adage that the dose makes the poison is definitely true. And so for some organisms, if you have a little bit of a compound, it's good. And if you have a lot of it, it's bad. And so when organisms are eating, they're facing this environment where they have to balance nutritional value, medicinal value, and toxicity as they make choices, or at least as they ingest, even if they're not thinking the way we might. And so this is the environment that they're facing as they forage. So I mentioned that sometimes they can use foods as medicines. This is something that's been studied a lot in terrestrial systems, or primarily in terrestrial systems. And so we've learned that in a wide variety of animal systems, organisms can use their food as medicines. And when I first started hearing about this, I assumed it would be like vertebrates, maybe like primates, but actually a wide range of animals can do this. So as one example, Drosophila fruit flies, if they see a parasitoid, and it doesn't even have to be right then, it could be like days earlier, they will lay their eggs in foods that are higher in ethanol concentration. And so ethanol isn't great for Drosophila, but it's really bad for their parasitoids. And so if there are parasitoids around, it's beneficial to them to lay their eggs in high ethanol foods. So animals are out there and they're medicating themselves and they're medicating their offspring. So as I mentioned, this is something that's been primarily studied in terrestrial systems, and this is a bit surprising because we know that aquatic systems have this incredible diversity of primary producers and that they produce a dazzling array of secondary metabolites. And so it's a little surprising then that this hasn't really received a lot of attention in aquatic environments. But before I go into that more, I want to make sure that we're all on the same page. So I just talked about phytoplankton and secondary metabolites, and I want to make sure we're all on the same page with those. Okay, so phytoplankton I mentioned a little bit briefly before, so I guess it's a little washed out here. But so there are some, the things that we think of as algae are all phytoplankton. There are also things that are blue-green algae that are actually bacteria, and we'll get to them a little more. But so they're the primary producers that are at the base of the food web. And again, the rest of the food web then obviously relies upon them. And I mentioned that there are different types of phytoplankton. For the lake systems that I'll be talking about, we can generally think of two main groups of phytoplankton for the purposes of this story. So there are the green algae, which we tend to think of as nutritious and things that don't make a lot of toxins and are generally like good foods that you might want to eat if you were an herbivore. And then there are cyanobacteria, which used to be called blue-green algae. Now they're referred to as cyanobacteria. And these are things that often end up forming blooms, especially in high nutrient conditions. Some of them, not all of them, but some of them produce toxins, and so they're also notable in aquatic systems for that reason. And I'll come back to blooms in a second, but I want to introduce the other term that I mentioned a couple of slides ago, which is secondary metabolites. So secondary metabolites are compounds that have no obvious primary role in metabolism, but they have some other role. So they're often things that help deal with some environmental stress. And so often that environmental stress might be consumption by herbivores, but there are other things. Secondary metabolites can be used for communication. They serve various functions. And then this is distinct from primary metabolites, which are things that are involved in the basic metabolism of primary producers. So all primary producers have those. With secondary metabolites, there's variation in whether they're produced and how much they're produced. So Mark Hunter has a great book on the phytochemical landscape, which is what that above quote is from. And then, as I said, he was the co-advisor for Crystal. And so he's done a lot of work on secondary metabolites. And so he notes in his book that the amounts of caffeine and theobromine that he consumes each day through coffee and chocolate would kill most organisms. And Mark does indeed consume really impressive amounts of caffeine. This, by the way, a friend of mine texted me this a couple of weeks ago. It looks like a Daphnia in her latte foam. And I love that she thought of me when she saw that. So the point about that is that these are not compounds that are inherently good or bad, right? These primary producers produce them because they're beneficial to them, at least in some circumstances. And then the effects they have on other organisms are really variable. So we really like the effects of caffeine and theobromine when we eat them, but they're toxic to most animals, right? So the effects are very much dependent on the organism and on the context. Okay, so these are things that I advise consuming at least sometimes. Microcystin over here is something I definitely do not advise consuming. And this is a secondary metabolite that's produced by Microcystis, which is a common cyanobacterium, and you'll hear about it repeatedly throughout this talk. So this is a picture from a pond that I used to study in Atlanta, which looks like this big swirl of green. And so microcystin is a really powerful toxin. And it's one, so now trying to tie all this together, I mentioned algal blooms before. So sometimes we end up with these primary producers, our phytoplankton, sort of out of control in our systems, and we can end up with a lot of these secondary metabolites. So this is a picture of Lake Erie. So this is shocking, right? So one of my grad students is from Utah, and she was like, yeah, when I moved here, algal blooms, it was like, so who cares? Like, you've got a lot of salad, you know? When you see Lake Erie looking like that, you realize that it's a problem, especially if you know that that is an organism that can produce toxins. I think it's ill-advised that that person is holding this big, mess of microcystis in their hands. And Toledo had to actually shut down its water supply for several days in 2014 because of a microcystis bloom at the intake pipe for their city water. So this is something that's become a big problem in the Midwest. Okay, so now with hopefully we're all on the same page in terms of phytoplankton and algal blooms and secondary metabolites. So coming back to this idea that we were really surprised by how little work had been done in aquatic systems looking at the effects of diets and secondary metabolites on host-parasite interactions, because we know from terrestrial systems that they can potentially have really big effects. And something that's especially notable to me is that these secondary metabolites are often studied as anti-grazing defenses. So microcystin is something that people who work on Daphnia studied for decades and generally didn't find much of an effect. And it was like, what's going on? And then there was a study done about 10 years ago that found that the ability to produce microcystin evolved before metazoans. So at least initially, this did not evolve as an anti-herbivory defense because there were no animals out there grazing on them. And so this got us thinking that maybe it evolved because it had antimicrobial properties. And if so, maybe those antimicrobial properties might be relevant for host-parasite interactions now. And so if we come back to our interest in diets and disease and thinking about how organisms are navigating this environment in which they have to obtain enough nutrition and maybe they have to medicate themselves, but they also don't want to consume too much of a toxin, what we were generally thinking is that if you have an environment that doesn't have any parasites around, you just have your herbivore and you have your primary producers, maybe our Daphnia over there on the side would want to choose what we think of as our nutritious foods, our green algae that are at the top of our menu. But again, a lot of these studies have considered just herbivores and primary producers, and they haven't considered the larger food web. And the larger food web always matters. That's like the one unifying theme of all of the research I've done, right? The larger food web is going to have an effect. So if we add on these parasites, which unfortunately they're not showing up well with this projector, but if we consider that there are parasites in the environment, it might be that in some cases for the Daphnia, the nutritious food isn't actually the best. Maybe this food we think of as being not nutritious might actually be worth consuming because some of those secondary metabolites might be useful, right? So they might be able to sort of medicate themselves. But also there's the whole idea of starving a cold. So it might be that having a less nutritious food is helpful if you have parasites around because then it gives fewer resources to the parasites to steal for them to use for their own fitness. So there might be multiple things going on there. And when I mentioned that these toxins might have medicinal effects, I think it's also worth noting that we already use a lot of the secondary metabolites that are produced by primary producers for ourselves. So artemisinin is an antimalarial that comes from plants, right? And so we use this ourselves. Okay, so for Crystal's work, part of what she was interested in looking at is whether diet influenced the outcome of host-parasite interactions and whether phytoplankton toxins can act as antibacterial and or antifungal medications in Daphnia. So by antibacterial, I mean effective against bacteria, right? So antibiotics. Okay, and so then coming back to the study system, so the host is Daphnia dentifera. It's a very common species in stratified lakes in the Midwest. It's usually one of the dominant, if not the dominant, species in those lakes. And then there are two parasites that we've worked on a lot that are pretty common in our system. So they're ecologically important, and they're also ones that we can work with in the lab. So over on the right, there's a fungal parasite known as Mechnicoia bicuspidata. I realize that Latin names are almost impossible to remember the first time you hear them, so I'm going to try to refer to it as the fungus the whole time. And then there's Pastoria ramosa, which I will try to refer to as the bacterium throughout this talk, just to try to make it a little easier. And so if you look at if you look under a compound microscope or an electron microscope, they look very different from one another. So the bacterial parasite looks like these sunny side up eggs. And the fungal parasite has these needle-shaped spores that actually completely fill the body cavity. So this is a scanning electron micrograph where I've sliced through the carapace, the outer part of the Daphnia's body, and you're looking into her body cavity, and it is entirely fungal spores, which does not seem very pleasant to me. And this is what they would look like under a dissecting scope with regular bright field microscopy. They look fairly similar at this level, but you actually can tell the difference even under a dissecting scope. Okay, and then if you were a Daphnia, I would not advise getting either of these parasites. So they're both very highly virulent. So if we look at the number of offspring produced, uninfected Daphnia produce many more offspring than ones infected with the fungus or with the bacterium. The bacterium is interesting because it actually doesn't have a significant effect on lifespan. It actually tends towards increasing it. So it's a castrating parasite. It shuts off reproduction, induces gigantism in the host, and then just turns the host into a parasite factory, which is pretty cool. But so I guess not if you're the Daphnia, but so both of these parasites are really highly virulent. And so again, part of why we're interested in them is we think they're ecologically important, and we're interested in understanding their roles out in nature. Okay, so for Crystal's experiment, what she did, a very coarse overview of the methods, was she took individual Daphnia, she put them in beakers, and then she exposed them either to the fungus, to the bacterium, or the lucky ones got neither parasite. And then there were 9 phytoplankton diet treatments. So there were 8 different species of phytoplankton, and I'll introduce those on the next slide. And then one of the species didn't make as much toxin in our lab cultures as we expected it to, so we added some more toxin to make it more realistic, more like what would happen out in nature. And then she tracked whether animals became infected and then how many spores were produced at the end of an infection. Okay, so here are the different diet treatments. So there were 5 different species of green algae for the aquatic ecologists in the group. They were Chlorella, Incastrodesmus, Eleutheryx, Chlamydomonas, and Cynodesmus. And then there were 3 species of Cyanobacteria. All of them are known to produce toxins, so Anabina, Microcystis, and Nodularia. And then again, our initial analyses showed the Microcystis wasn't making much microcystin. It was making some, but not as much as we thought it would. And so we spiked it with additional microcystin to get it to the levels we would expect to see during a bloom. So we weren't trying to make it unrealistic. We were just trying to get a little higher, but still ecologically relevant. And I will use the green and blue color coding throughout because, again, I don't expect you to remember Latin names that I just threw at you for the first time. Okay, so looking at this question of how diet influences infection prevalence and spore yield. So I mentioned before that we can tell if they're infected by looking under the scope because they're transparent. This is very convenient. So we often do this using dark field microscopy. So the animal on the left is uninfected and the animal on the right is infected, and that's why she's so much brighter white. And in this one, you could actually, if it was a little bigger, you would be able to see the individual needles in the head. So that helps you tell the 2 species apart. And then in terms of spore yield, we do this. We grind up the animal in a known volume of water, and then we use a hemocytometer, which is a special kind of slide that has this grid on it that allows us to count the spores. And this is what it looks like under the scope. So each of these here is a spore of the fungal parasite. So we can then calculate how many were in the host's body. Okay, and so then looking at the data, just to make sure everyone's oriented. So there will be dots for each of the different treatments, so, and all the blue ones are the blue-green algae. All the green ones are the green algae. And then proportion infected is on the y-axis. So if you're at the bottom, none of the animals became infected. If you're at the top, all of the animals became infected. There were 10 replicates for each of our treatment combinations. Okay, and so for the fungal parasite, there was variation in among the different diets and how infected they became. But what stood out the most to us is that 3 different diets completely prevented infection by the fungal parasite. So this was a very strong effect. Interestingly, for the bacterium, there was no significant influence of diet on whether they became infected. Like, that's a pretty boring result there. If we look at spore yield, it's actually the reverse. So for spore yield, so here we don't have as many treatments because we can't measure spores if they didn't become infected. So we've lost some treatments at the top. But diet did not influence spore yield for the fungal parasite, but it influenced it significantly for the bacterial parasite. And what was interesting is that spore yield was actually higher on the cyanobacterial diets, which we think of as being lower quality. So this was the opposite direction of what we expected would happen. In general, I am very bad at predicting what will happen in experiments. And then those were focused on parasite fitness, but if you're wondering about host fitness, so I mentioned that we think of green algae as being more nutritious than the cyanobacteria, and the Daphnia tell us the same thing. So if we look at their offspring production on the x-axis, it's notably lower for the cyanobacterial treatments than it is for the green algae. They like the green algae a lot more. If we look then at when they're infected, at what happens, they still produce more offspring. I'm sorry this is so washed out. They still produce more offspring on the green algae, but they take a big hit in their overall fecundity, right? So they're doing a little better, but not nearly as big a difference as when they're uninfected. And then for the bacterial parasite, there ends up being a lot of variation. And really, this is variation in whether they get sterilized by the pathogen or not. And there's probably a lot of interesting stuff to follow up on with that. Okay, so yes, diet definitely had a strong influence on fungal infections, on whether they became infected. It also had an influence on bacterial infections in terms of how much the parasite grew up within the host. So if we look then at whether phytoplankton toxins might act as antibacterial or antifungal medications, I want to come back to a slide that I showed you already and spend more time pointing out what's on the x-axis here. So this red arrow indicates the treatment that had the microcystis plus additional microcystin added to it. And right next to it is the treatment that just had the microcystis. And so what's really notable is that when we added a little extra microcystin, so that's the only difference between these two treatments, we had lower levels of infection. So this suggests antifungal properties of microcystin. The other cyanobacteria were interesting too. So Anabina was one of the other cyanobacteria that we looked at. It produces anatoxin, which I'll discuss a little bit more. But first, so remember, Anabina completely prevented fungal infection. So it was another one that prevented these infections. So what's really interesting to me is that it seems that anatoxin is induced when you have the fungal parasite in the water. So I actually lied before when I told you there were 3 infection treatments. There were actually 4, and I've just left one of them out until now for simplicity. But so there was the control that had no parasite, there was the treatment with just the fungus, the treatment with just the bacterium, and then a treatment where we added both of them. And so both the treatment with just the fungus and the treatment with both of them had significantly higher antitoxin concentrations than either of the other two treatments. So this is notable for a few reasons, including some of our lakes, our study lakes, have Anabina blooms. We see them. So this is a picture I took of a colony that was produced during a bloom during one of our normal field sampling. And antitoxin might not sound particularly exciting to you, but its other name is very fast death factor, which is amazing to me that that's its other name. So you can imagine that you wouldn't want to have more very fast death factor in a lake, right? And so we know we have blooms of this, and then now we know that this fungal parasite seems to be inducing it. So we might see some variation. And again, this is, these lakes are the sources of drinking water for some communities, so this might be something that's worth thinking about a little bit more. So I mentioned before that antitoxin or Anabina prevented fungal infections. Antitoxin seems to reduce spore yield in Daphnia when they're infected with the bacterial parasite. So this is the antitoxin concentration on the x-axis and the spore yield on the y-axis, and there's a pretty clear negative relationship there. So it seems like there might also be some antibacterial properties of antitoxin, and that's another thing we plan on following up on. Okay, and then our third cyanobacterium that we looked at was Nodularia, and it produces nodularin. And this one is interesting because there were the effects of nodularia or nodularin seem to be like a classic example of medicinal properties of a compound. So if you look at the uninfected animals, so when nodularin concentration increases, their offspring production decreases really notably. Whereas if we look in the fungal infected animals, they actually have a significant increase in their offspring production with increasing nodularin. And this is common with medications. I don't take antibiotics all the time because antibiotics would have negative impacts on me if I'm not infected. But if I'm infected with some nasty bacterial pathogen, I'll take those hits that come with taking the antibiotic because it's more than offset by the effect that it has on the bacteria. So this is a very clear example of medicinal properties of nodularin. And so if we summarize the results, diet had a strong influence on fungal infections, and then parasite fitness decreases and host fitness increases in the presence of toxins. So it does seem like there are medicinal properties of some of these compounds. And to be clear, we're not saying that the Daphnia are self-medicating, right? So I used this example with the Drosophila at the beginning. We have no evidence yet that Daphnia are self-medicating. This is an obvious next step, and we're starting to work on that. So we don't know if they're actively choosing what they eat. We tend to think of Daphnia as these cows that are just mindlessly grazing, but there is some evidence that they can be selective. And so now we're starting to do work to see if they do alter, if they can selectively feed, and if so, whether the selectivity of what they choose varies depending on whether there are parasites in the environment. Okay, so at the beginning of the talk, I talked about how foods combine nutritional value and medicinal value and maybe toxicity. And so, and I also mentioned how in aquatic environments in particular, we've tended to focus on nutritional value. But so what we're starting to do is to understand how these different foods might have different values, but also that might indicate that might vary with the different parasites, and it seems to differ with parasite identity. So this is a bit of a mess, and we're going to have to spend some time trying to figure out why we see one effect for the fungal parasite and a different effect for the bacterial parasite. Does that hold more generally for fungi versus bacteria? We don't know any of that yet, and that's stuff we plan on looking at. But it's definitely worth keeping in mind that when these organisms consume food, it's providing more than just nutrition, and also the net effect of that will depend on what else is going on in the food web. Also, I didn't emphasize before, but I'm going to start talking more about Chlorella. So Chlorella is like the magic algae in this experiment. If you were a Daphnia, I would tell you to eat a lot of Chlorella. And actually, you don't have to be a Daphnia. You can find this, like we found it in a face wash. My grad student sent a picture of that. They sell it in health food drinks and stuff like that. But it's a green algae that was really nutritious to the Daphnia. They did really well on it when they were uninfected. They made lots of babies. It also prevented the fungal infections. So we're not sure what's going on with that, but it seems like Chlorella is a really interesting one to follow up on. And so the previous slide had sort of the cartoon version where we're thinking about these circles. But I think a lot of the results also can be summarized by this picture. So these are two Daphnia who are the same genotype and the same age. And the one on the top, it's a little hard to see. Her gut is green, and this one is more of a blue-green. So they've been feeding on different algae. This one is notably shorter. If you look at her length, she's not as long as the other one. She only has one baby in her brood chamber instead of a whole ton of babies in her brood chamber. But she's not infected. And that one is. She has all this, the fungal parasite growing around her heart. So there are definitely some interesting trade-offs going on for these organisms in terms of what foods are lead to the highest fitness. Also, Daphnia are pretty. So Crystal's results were really striking. So there were these 3 phytoplankton diets that completely prevented infections. And as I said, we did this work because we thought there might be some effect, but it was a stronger effect than we expected. And so the one of the obvious next steps then was for Crystal to take some of the extracts from her cultures and see if they have broader antifungal properties. And before I get into that more, I thought it would be worth explaining why we might care about whether something has antifungal properties. So we tend not to think about fungal diseases as diseases that are important in humans, but each year 1.5 million people die from fungal diseases. So that's 3 times the number that die from breast cancer. And I mentioned this to my hairstylist at some point last year, and she was like, "It needs a ribbon. It needs a month." She got all worked up. Right? So these fungal diseases are important, especially globally, but they're not ones that we tend to hear about. And part of it is that for healthy people, fungal diseases are a nuisance. It's like athlete's foot or something that's annoying, but not going to actually influence your fitness. But for people who have another condition, so maybe HIV or cancer or cystic fibrosis, fungal diseases can be really, really problematic. Or also for people who have surgical procedures, or certain types of medical devices. So we don't like to think about it, but fungi are floating around in the air. And so if someone's, say, having open heart surgery, sometimes a fungal spore can get into their body. And if it's in their bloodstream, then it can be fatal. And so this is a picture of one kind of fungus, Candida, that is responsible for 40% of hospital bloodstream infections. So just this one fungus causes quite a lot of hospital bloodstream infections. And then fungal diseases are really, really important crop diseases. So more than 2/3 of all crop diseases worldwide are caused by fungal pathogens. And then fungi have emerged as a really important conservation concern. So the chytrid fungus that swept through amphibian populations globally has really decimated many of them. And then now there's a salamander, a close relative that's infecting salamanders. There's a fungus that's emerging in snakes. There's a fungal pathogen that's emerged in bats. And so fungi are emerging as something that are really important pathogens from a conservation perspective. And there is some sense that fungal diseases might be on the rise and that some of this might relate to global change. And this is actually some of the work we do in my lab is looking at how different aspects of global change might be influencing parasites and who we see where and how severe the diseases are. Okay, but I mentioned that we were interested in looking for antifungals before, and we have very, very few antifungals now. So you've probably heard that we have this crisis with antibiotics where the bacteria evolve resistance to them. There's a similar thing where fungi evolve resistance to the antifungals, but we're starting with even fewer antifungals than we have antibiotics. And some of the antifungals we have are really marginal as medicines. They're really toxic, and you wouldn't give them to someone unless they were going to die of the fungal infection otherwise. So we're really, really limited in what we have now. So we really need new antifungals, and to me it's really exciting to think that we might be able to find some of them just by looking in lakes. And so Crystal went in the fall to a collaborator's lab at Georgia Tech where she tested the activity of some of her compounds against a fungal pathogen of humans. And this initial screening found, so this is for Chlorella, the extract from her Chlorella cultures was strongly inhibitory against a drug-resistant human fungal pathogen at concentrations that are reasonable. And to me, the Chlorella result is the most exciting because with microcystis, if microcystin is effective against antifungals, it's going to take a lot of work to turn microcystin into a human medication, right? It's a really powerful toxin to vertebrates. But Chlorella, as I said, is something you could find at your local health food store. And so this has much more promise. So we're pretty excited about that result. And so Crystal is going to work this summer. She's going to go work out of the lab of a medicinal chemist who has a really large suite of human and agricultural pathogens that Crystal can screen these compounds against, and she can do the full drug discovery project. And so my father is a retired New York City firefighter, and when I talk about my research with him, he'll often say, but how is this going to help people? And I've tended to be like, probably not. And he wants a really direct application to people. But again, I tended to think, probably not going to be there. But it seems like I was wrong. There might actually be a direct application there that I didn't see coming. Daphnia might be able to teach us how to fight fungal infections in people. And so as I said before, we started studying Daphnia because they're really key players in lake food webs. But when we studied Daphnia and their parasites, we found these compounds that prevented fungal infections in Daphnia. And then now in our initial screens against fungal pathogens that infect humans, they seem to have some antifungal properties there. So to me, this is really incredibly exciting. And it's also, if you asked me 2 years ago what work we would be doing, I would never have listed drug discovery. This would be a project that we would be working on in my lab. But this is how basic research works. You're working on a topic that seemingly has no direct relevance to people, and then you have some unexpected result, there's some unexpected breakthrough, and you end up with something that does have an application to people. And so I also wanted to note that this isn't just a story about the value of basic research. To me, it's also a story about the importance of diversity in science. So Crystal, who led this work, was in a federally supported program that aims to increase the diversity in ecology and evolutionary biology. And in my opinion, Crystal is addressing questions that people didn't think to ask before, and she brings really interesting new insights to them. And she's getting really incredibly exciting results. So it's definitely too early to say whether Crystal's work will lead to the next big drug to fight fungal infections in people, but it's already abundantly clear that science is stronger because of Crystal being in it. And so Dr. Mark Schlissel is the president of the University of Michigan, and to paraphrase him, he said something like, talent is evenly distributed in society, but at present opportunity is not. And I think it's really important to note that science will progress further and faster if people from all backgrounds are able to contribute their talents to science. Thank you. Yeah.
Megan, your two primary treatments for greens and blue greens, do you consider diatoms? So why or why not?
Yeah, so we decided not to use diatoms for this. I think it would be really interesting to look at that. We don't have a good sense for our lakes in Michigan. So one of the things we're planning on doing is getting a better sense of what the phytoplankton assemblages are at different times of the year. These disease outbreaks tend to be happening in late summer and autumn. And so There are certainly a lot of cyanobacteria around. There are certainly a lot of greens around. We don't see as many diatoms, but we're not really sampling in a way designed to catch the diatoms. And we certainly have seen when we've set up these whole water column enclosures, we get diatoms growing on the sides of the bag, right? So they're around, but we're probably missing them with our sampling.
They just tend to be much higher food quality than
Yeah.
the greens.
Yes. Yeah, I'm sure the Daphnia would be super happy with them. They're already pretty happy with the greens. Yeah.
I'll probably make a mess of this, but I'm going to ask anyway. So you showed the infection rates, the survival rates, and then the amount of offspring each individual produced. Have you tried essentially sort of multiplying all those together to get an estimate of like lifetime fitness?
Yeah, well, so we definitely multiply the infection prevalence and the spore yield together. And we've decided to call that transmission potential. We just made that up. But yeah, that's like definitely the parasite fitness is a product of that. For the Daphnia, I think the offspring production is the measure of fitness, and it's already taking into account whether the parasite infected and how much it's growing. We did see, I didn't mention this, but what were not on that figure were individuals that were exposed to the parasite but that did not become visibly infected. And they do pay a cost. They have lower fecundity. And we're just starting to learn more about, it does seem like the parasite gets in and then the Daphnia fights it off in some cases. But yeah, so for the Daphnia, and also with the stuff with food quality, like Crystal at first was thinking about doing like C to N to P and measuring fatty acids. And I was like, the Daphnia are telling us what the quality is. Like we could measure the chemistry, but the best indicator of what the quality of the food is, as far as we can tell, is the fecundity of the Daphnia. Yeah. So for the parasite, though, I think definitely multiplying them together is an important part of their fitness. Yeah.
So you mentioned in the beginning in your introduction that the cyanos that have these secondary metabolites, their production of the metabolites evolved prior to the metazoans, right?
Yep.
So you're showing this effect of antifungal properties in the metazoans. So do they have antifungal properties for the cyanobacteria themselves? Like, so what is the relationship there?
Sorry. So does who have anti?
So I guess my question is, why do you think the cyanobacteria have those secondary metabolites in the first place?
Yeah. I mean, our guess is probably to deal with other bacteria was probably the initial. And so if I had to guess at the start, I would have said there would be stronger effects on the bacterial parasite. But it's clear they've been co-opted for other uses. And yeah, so they did have some effects on the bacteria, but the dominant effects were on the fungi. And yeah, so our initial motivation for going into this was this whole like, oh, they must have evolved as antibacterial, but they seem to have, for us, for these two parasites at least, they seem to have stronger antifungal properties. Yeah.
Okay, well, Susanna took off a little early to, ice the drinks. So she asked me to remind you, 33 Winding Way. Let's just go up Center Street up to Cliffside, and you'll find your way there. And hope you all can join us. And thank